people are registered, sir.
Great. That shows your audience base worldwide.
Yes, sir. Most of the Asian and Africans, actually.
You are reaching out to a larger audience.
Yes, this is our intention also. They should know about plant scientist people and about plant science.
Yeah.
Very
We are live.
Okay. Few people are working on plant, and that is the reason we are.
I will start recording when you say.
Yes, just a minute. I just wanted to check that. Yes, we are live. Okay. Okay, now we should start.
Okay, so I will record on this computer.
Welcome everyone and Happy New Year. Sorry, in 2023 January we can't go for any webinar. We have some work to do, so we can't arrange that time. And this is our first webinar in 2023. Welcome all of you, and this is our 54th number webinar, and I'm very happy that today Dr. Swamy from IRRI there. I know him about for his work. He is a very good and renowned scientist, specifically when it's come to the biofortifications of rice, specifically for the golden rice. His work in golden rice is very commendable. Many times I cite him on my publications, and I realized that how much important and how much of love he has on the rice biofortifications and rice work. Welcome, sir, to the Bioingene webinar. I give a certificate application link during the end of the webinar, so keep patience. This link has to be submitted within 4 or 5 hours, and we will distribute a participation certificate through Bioingene website. It will be available on 28th of this February. I request Shoma to introduce Bioingene a little bit, give the instructions about certificate, and introduce our speakers. Thanks everyone for coming and joining to this webinar. Let's start. Shoma, please.
Okay. Hello, everyone. Welcome to another Bioingene webinar. Bioingene is a not-for-profit organization created to promote plant science research worldwide. The webinar series has been designed to build a platform from where plant scientists can present their research to the world. We hope future scientists can gain perspective and inspiration by listening to the esteemed plant researchers talk about their scientific accomplishments and their thoughts on the future of plant sciences. We are grateful that many renowned scientists have accepted our invitation to share their research insights with us. We are also thankful to you, the viewers of our Bioingene family, for your interest in Bioingene webinars, your constant support and appreciations. We have received a huge response for today's webinar. You can register for the webinars through our website, bioingene.com, and our social media pages, and live view on our YouTube channel. Video recordings of all our webinars are available on our YouTube channel. After attending today's talk, you can apply for a certificate of participation via the feedback link, which will be provided after the presentation. You can collect your participation certificates from our website after 2 to 3 days. Bioingene does not send certificates through email. Please type in all your questions related to today's talk in the YouTube chat box for the Q&A session. The topic for today's webinar is Recent Advances in Breeding for Healthier Rice by Dr. B.P. Mallikarjuna Swamy. He is a Senior Scientist 1, Rice Breeding and Biofortification at IRRI, Philippines. Dr. Swamy successfully completed his doctoral degree in genetics in India in 2009. His PhD work resulted in mapping of major QTLs for yield and related traits, registered novel germplasm, and released a high-yielding rice variety. His interest in the effects of climate change on food security and widespread malnutrition led him to work on the complex and challenging task of developing climate resilient and nutritious rice varieties. Later in the same year, he moved to the International Rice Research Institute as a postdoc fellow in drought molecular breeding, wherein he mapped major QTLs for grain yield under drought and successfully pyramided them into popular rice varieties along with major QTLs for biotic and abiotic stresses. He significantly contributed to the genetics of drought tolerance in rice through meta-analysis and identification of candidate genes. His works were well recognized at IRRI and worldwide, and he was appointed as lead breeder for healthier rice development in 2013. He has been playing a pivotal role in developing healthier rice, and major milestones have been achieved in developing golden rice and micronutrient-rich rice varieties. He is also a prolific writer, published in high-impact journals and actively involved in disseminating knowledge of health and nutrition and training future researchers. He is an active member of plant science societies and editorial board member of scientific journals. We are highly honored to have him with us today on the Bioingene Global platform. I now request Dr. Swamy to please begin his talk.
Thank you very much, the Bioingene team, and I must congratulate you people. Your team is really doing a great job, and you are reaching out to so many researchers and providing them with updates of scientific updates on research in plant sciences, and it's benefiting a lot of people. So, and I also thank you for this invite and to be part of this today's seminar. And I would like to interact with so many students. You know, I saw the list. I saw so many students, researchers across the world. So it's also kind of a great opportunity for me to interact with them and also learn from you people. Learning never stops. It's also kind of learning from you people. So thank you very much. Yeah, let me start. Can you see my slides?
Yes,
Yes.
they have started, yes.
Okay.
Yes.
Okay, thank you. And for the last 12 years, I think almost 12 years, I have been working on developing nutritious rice varieties, or also called as healthy rice varieties. So a lot of progress has happened over these many years. So I will be giving you some updates of our work. So can you hear me?
Yes,
Is it
sir.
audible? Yeah.
Yes, sir.
Yeah. As you all know, always there was a lot of interest and discussions around food security. We have this many population, there will be projected growth in numbers of this many numbers of people by these many years. We need to provide food to all these people. So there will be always a lot of pressure on the agriculture sector to produce more with less land, labor, with less resources. There is always a tremendous pressure on the agriculture sector to provide food, because food is basic thing. You need food to live, to be active. You need food. So that's the basic thing for life. So you can see yourself, almost 8.5 billion people will be there by 2030, and most of this increase is going to be happening in Asia and Africa. And the land resources are limited, and we need to produce more with every unit of resources. As I mentioned earlier, with less land, labor, water, with everything less, we need to produce more. Means our productivity has to go up. And that was the main focus all these years, food security. And still it remains to be one of the important focus for agriculture sector. or the plant science sector. At the same time, in the last 1 or 2 decades, there is a lot of interest on nutritional security as well. It's not only just eating or living, eating nutritious food and leading a healthy life. That's the most important thing. It's not just living. We need to live a healthy life, to be happy, to be active, to enjoy life, social life, everything. You need to be healthy first. So it's not just about providing food. It's also about providing nutritious food. As you know, almost half of the world population is in one or the other way, they are micronutrient deficient in the developing world. On the other hand, in the developed world, obesity and the overgrowth is a major problem. So it's a kind of double burden. On the one side, overnutrition, and the other part of the world, less nutrition. So this micronutrient deficiency is also called as hidden hunger. It's a huge health problem because most of the people in the developing world, we depend on staple cereals like rice, wheat, maize, some cassava, potato. So we depend on these staple crops for our daily energy needs. But these energy foods, they are not able to provide the amount of micronutrient that we require. On the other side, the people in the developing world, they don't have resources to diversify their food, to get nutrition either through diversifying their food or by eating fruits, vegetables, meat. So they may not be able to get the recommended dietary allowance of the micronutrients. So with the as we started achieving food security, nutritional security is gaining importance. So a lot of interest in developing crop varieties with nutritional value. And considering the importance of nutrition in human health, United Nations declared improved nutrition as one of the sustainable development goals. So that itself shows the importance that has been given for improving the nutrition of the people. So now we can say both food and nutrition security are equally important. It's not just food security, nutrition security also very important. That's why there is a lot of interest from the global agencies. They are investing a lot in developing nutritious crop varieties. So these are some of the important nutritional and health problems globally. If you can see this slide, vitamin A deficiency, zinc deficiency, iron deficiency, and protein energy malnutrition. On the other side, if you see health issues, diabetes, heavy metal toxicities, cardiovascular diseases, these are some stress. These are some of the major health-related issues globally. And especially children, pregnant and lactating women are highly prone to micronutrient deficiencies because the growth and development, and pregnant and lactating women, those who are giving birth to a baby, they need more nutrients. So it's our duty to provide nutrition to these people to make them healthy. So in addition to providing food. So as you all know, since people eat the staple food for their energy every day, for example, in Asia, we eat rice 3 times a day. So if you put nutrients to these staple crops, indirectly they get nutrients. So every time you need not to go and tell them that to take the nutrients. So you put nutrients to the food they daily eat, so they get the nutrients. That's the main objective of this biofortification. But as you all know, during Green Revolution, we could able to develop high-yielding rice varieties, but that was not commensurate with increase in the nutritional value. At the same time, our eating habit, the behavioral changes happened in terms of the way we consume rice. We polish it. We polish and we consume polished rice. So most of the nutrients will be lost. So the purpose of this biofortification is to put these nutrients in the edible portion in the endosperm, so that even after polishing, the nutrients are remained intact in the rice, in the endosperm, and once they consume that, so they get nutrients. That's the main idea around the biofortification. So biofortification is not the only way to get the nutrients, as I mentioned earlier. You can change your diet, you can diversify your plate with other foods, vegetables, roots, fruits. You can change your diet, diversify your diet. You can get nutrients. And through supplementation also you can get your nutrients. Like you can get tablets, and you can get fortified foods. You can get nutrients. Biofortification. So the one we do, what we do here is that using the breeding and agronomic approaches or the genetic engineering or genome editing approaches, we increase the nutritional value of crops by increasing the nutrient content of the beneficial elements like iron and zinc, vitamin A, like that. And also by enriching the compounds, promoter compounds, which helps in the absorption and bioavailability of the iron and zinc and other mineral elements, and also by reducing antinutrients. These antinutrients obstruct the absorption of the mineral elements like a phytate content. So we bring down those compounds in the edible portion of the crops, so that people, when they eat these biofortified crops, they can get more nutrition. And you can see yourself that on an average, the recommended daily intake of vitamin A is around 600 microgram, and iron and zinc is around 15 milligram per day. But most of the modern rice varieties, where almost 90% of the rice is produced and consumed in Asia, and also rice is becoming a major diet in Latin America and Africa as well. But most of the high-yielding modern rice varieties, they have 2 to 3 ppm of iron in the polished rice, also called millet rice. And the zinc, it has around 12 to 16 ppm, and there is no variability for carotene or vitamin A in the rice endosperm. So considering all these approaches, you can diversify your diet, or you can go for supplementation or biofortification. Biofortification is the most sustainable, targeted, and cost-effective approach, because once you make an investment and develop rice varieties, once it reaches the farmer, you can continuously grow and eat them. You need not invest every time. On the other side, for supplementation, every season you need to, government has to invest money, and you need to produce those tablets, syrups, and the fortified foods, and then you have to take it and make it available to the targeted groups in the even rural areas or wherever they are. You need to go to them and make it available. It needs recurring investments. And change of diet, you can advocate, but if those are available and affordable, they can buy it and eat. So compared to other approaches, since biofortification varieties are once available, they can continuously grow them and eat it. It's the most sustainable and cheaper. It doesn't need any extra investment. So, and it will be like any other ordinary food. You are buying a rice and you buy a biofortified rice, it's almost same cost, but you get nutrition from the biofortified crops. At IRRI, we work on projects in terms of rice biofortification. We develop high zinc rice varieties by conventional breeding and also molecular marker-assisted backrest breeding program. And also we develop high iron zinc rice through transgenic and genome-edited approaches. And also we did a marker-assisted breeding program for integration of vitamin A or the golden rice gene into popular rice varieties of Asia. And also at IRRI we work on low glycemic index rice, also called as diabetic rice. And also we work on heirloom rice, also called as traditional land rices characterization. You know, these traditional land rices, they are a rich source of nutritional elements and health beneficial compounds. Heirloom rice is nothing but all our traditional germplasms. So, and also at IRRI we communicate through our channels dietary diversification, the importance of dietary diversification to get the nutrients. So they not only just eating rice, we tell them to diversify their food so that they can get more nutrients. And specifically, so on zinc biofortification breeding, my team, we work on zinc biofortification. So I will give you a little bit more information on zinc biofortification. So you can see if there is any zinc deficiency, you can see there are a lot of health issues. Diarrhea and stunted growth are highly visible. So in addition to that, all immunity related issues, growth and developmental issues, reproduction related issues, so many issues are associated with zinc deficiency. People may ask, why zinc? Why not iron? and both are given equal importance. Then people also ask why they are equally treated, because iron is more needed. So zinc is an important catalytic agent for more than 300 enzymes in the body. And also you might have heard and seen, more than 3000 proteins. For their structure and functional stability, zinc is very important in the human body. So that's why it's very important. Even for the absorption of iron, zinc is required, because zinc is required for most of the enzymatic activities. Even you may take a lot of iron supplement. So if zinc is not there, it will not be taken up so easily and quickly. So that's why whenever people go to a doctor or a physician, they give both iron. So at IRRI, we are working on development of high zinc rice varieties. And for any breeding program, you need to have a target. So we need to have a target. That target is set by the nutritionist and physiologist. So looking into how much of rice is consumed on an average per day, and how much of that zinc is removed when you wash it, cook it, and how much of that zinc when you consume it is readily bioavailable for the body. So looking into all those considerations, they have set a target of 28 ppm. And as I mentioned earlier, most of the modern rice varieties have 12 to 16 ppm. That is not enough to meet our recommended dietary intake that is needed on a daily basis. So our target is to increase the grain zinc content of modern rice varieties by additional 6 to 12 ppm. That means over the 16 ppm, we need to add 6 to 12 ppm. As you all know, the biological sciences, so all of a sudden you cannot make such a big increment. So it goes with small increments. Like in the first wave, we want to release rice varieties with 6 to 8 ppm, and in the second wave 8 to 12 ppm. In the third wave, rice varieties with more than 12 ppm over this basal level of 16 ppm. That's the target. So we already developed and released high zinc rice varieties from the first wave and second wave, and we have breeding lines with more than 12 ppm over this basal level at the testing level. So you can see, even though the average requirement is 15 milligrams, some need more, some need less. So depending upon their age, gender groups, and it varies. So you can see the zinc requirement of different age groups and gender. So this is the requirement. And if you look at the distribution of the grain zinc in the rice grain, most of the zinc is still, almost 60% of the zinc is still in the endosperm, and almost 40% is in the brawn and embryo. That means even you polish it, a significant amount of zinc is retained in the rice grain. And also there is a variability in the rice germplasm collection. As a plant scientist, you all know variability is must for any breeding program. You need to find a variability for any trait of your interest, whether it's for nutrition, whether it's for disease resistance, whether it's for abiotic stress. In the germplasm collection, you need to find variability for that. In terms of biotic stress, you need to find the resistant donors. You need to, for abiotic stress also for drought tolerance, for example, you need to screen them, identify the drought tolerant donors, then bring them into the breeding, and then you start the process of breeding. Similarly, for zinc, we need to identify high zinc donor lines and use it in the breeding program. And there is variability for zinc in the rice germplasm collection, so that we can do breeding for conventional breeding for high zinc improvement. On the other hand, for iron, most of the iron is in the brawn and embryo, so that will be lost when we polish it. Very little amount of iron is retained in the embryo, in the endosperm. So, and also not much variability with available information and whatever characterization has happened in the germplasm collection. So the best way for iron improvement is through transgenic or genome editing approaches. So for zinc, we do conventional breeding, marker-assisted breeding, and also genetic engineering or genome editing approaches for zinc. But for iron, we are doing genome editing and genetic engineering. Similarly, for vitamin A, there is no variability for vitamin A in the rice grain. So transgenic approach is the only way forward. So we apply transgenic and genome editing for iron, vitamin A, and both approaches for zinc improvement in the rice. And this is the scheme we follow at IRRI. We follow both conventional breeding and molecular assisted backcross breeding programs and GWAS and all the techniques for trait mapping and also transgenics. And finally, once the products are ready, we characterize them, we take it through varietal trials, and finally they will be tested in the national systems trials for varietal release, and finally they get them released for commercial cultivation and for consumption by the target groups. And if you look at the variation for grain zinc in the rice germplasm collection, you can see that this diversity diagram. Indica varieties are the one that we eat. So in this part of the world, in Southeast Asia and South Asia, so mostly we eat indica rice. If you look at the grain zinc content, indica are the lowest in terms of grain zinc in their endosperm compared to japonica, house, and aromatic. And house is genetically closer to indica, so we try to identify high zinc donor lines from the house subgroup. Before me joining, the breeding program was heavily based on using japonica. It's not that easier to do breeding between indica and japonica and to combine the traits. But house is genetically closer to indica, and house accessions are found to be rich in nutrients. So what I did when I joined, we collected almost 3,000 accessions of house from the IRRI gene bank. We characterized them. Finally, we selected 600 accessions, and we grouped them into two groups, 300 and 300, based on their maturity, flowering and maturity. And we evaluated them. And finally, we selected 40 best ones, which are good in terms of yield and also good in terms of zinc content. Because why? Because for a farmer, profit is most important. So yield is very, very important. You look at yield. Zinc is not visible for a farmer. For example, pest and disease is very visible, and you can see the impact of pest and disease. But for zinc, unless you take the grain and put it in the machine and measure it, you cannot see by bare eyes. So it's a kind of indirect benefit. So for health. So for a farmer, he has to adapt nutritious varieties. Means yield should be good, should be like any other high yielding variety that is available in the market. The interesting thing is that there is a relation between yield and the nutrient content. If you look at the literature, if you have searched through the literature, you see always there is a negative relation between yield and the nutrient content. If any variety is very high yielding, it will have some compensation in terms of nutrients. If it is low yielding, it has more nutrients. We call it as a concentration effect and dilution effect. But as a breeder, as a scientist, it's our job to find exceptions, means like high yielding genotype having high nutrients. So in house germplasm collection that we screened, we found accessions almost 40, which are having acceptable yield potential and very high levels of grain zinc content. And these 40 accessions, we grew them across several locations in the Philippines, because grain zinc is a quantitative trait. Its trait expression varies with environmental influence and soil conditions and climatic factors. So we want stable genotypes or the stable donor lines. So after testing those 40 genotypes, we selected the top 2 best, and we use these 2 accessions for our trait genetics and also for breeding program at IRRI. And they have almost 40 to 45 ppm of grain zinc content in the endosperm with acceptable yield potential. And always people ask a lot of questions. Everybody, those who are all involved in our rice science or the plant science, even plant engineers, they have their different opinions. So just to talk about the complexity of the genetic basis of grain zinc content, as I mentioned earlier, it's very highly complex. It's controlled by more than 80 genes and highly influenced by the environmental factors. And the final grain zinc content, in the grain, it's because of partly it's from the remobilization from the flag leaf and partly from the direct root uptake that makes the final grain zinc content. And people say you can apply zinc into the soil, so you can get high zinc rice varieties. That's not true. You can apply any amount of zinc to the soil. If the plant is not capable of releasing it and taking it up and loading into the grain, see, any amount of zinc that you dump to the soil will not make any difference to the grain zinc content. And also I mentioned dilution effect and concentration effect. So the yield has a relationship with the nutrient content. And also artificial zinc deficiency and natural zinc deficiency. Artificial zinc deficiency means there is a lot of zinc in the soil, but it's not readily available for the plant. So in such a situation, plant has to produce a lot of root exudates and make it available and translocate it and load into the grain. That needs energy, that process. So that will have kind of compensation on yield. And also remobilization, as I mentioned, if a plant is highly efficient in remobilization of zinc from the leaf, it leads to early senescence, means leaves turns yellow. So there will not be any more photosynthetic activity. The bottom 15 to 20% of the grains, they won't fill. It will have a yield penalty. So that is the interesting thing. A physiologist, we need a physiologist who can characterize that. Even flag leaf mobilizes its 90 to 95% of the zinc. Even with remaining 5%, it should continues to be green, continues to do photosynthesis, and continues to provide photosynthesis to the bottom portion of the grain. So that will be more helpful for a biofortification breeding program. And natural zinc deficiency, there is no zinc in the soil. Plant cannot do anything. You need to apply zinc to the soil. And also there are studies saying soil application and foliar spray. As I said, soil zinc application, you need a right genotype that has the ability to take it up, release it and take it up. Foliar spray, yes. If you spray during a flowering stage, grain filling and flowering stage, so it has 2 to 3 ppm increase in the grain zinc content. And I already mentioned remobilization versus yield penalty. And also irrigated, rain-fed and AWD, rice is cultivated in different ecosystems. You can prepare land, transplant it, and you make water standing water. And also in the rain-fed conditions, there will not be any standing water. In the AWD, alternatively, you wet the field and dry the field. So these are the different ecosystems. If you compare these ecosystems under irrigated conditions, the zinc availability is less compared to rain-fed and AWD. On the other hand, rain-fed and AWD conditions, there will be less yield compared to the irrigated conditions. So zinc is more available under aerobic conditions. And even rainfall, seasonal effects, soil factors, so many factors affect the grain zinc availability, uptake, and loading into the grains. And also zinc deficiency operates at the system level. I will explain you in the next slide. So overall, it's a combination of breeding, crop management, soil management, water management. Everything has to work together for a successful biofortification, zinc biofortification. And I was mentioning zinc deficiency operates at some level. Just to curiosity, one of my PhD scholars, I told him that to do this study. So just gave him a few bullet points. He's a very intelligent guy. And I told him what to do. He did everything. So he went to the Philippine Bureau of Soil Science. He collected the soil nutrient status, soil micronutrient status, and he went to field rice and he collected the rice crop nutrient status. And also he went to the Philippine Nutritional Institute and collected the human nutrition status across different provinces in the Philippines. Then he superimposed data, soil nutrient data, plant nutrient data status, and also human nutrient data status using the GIS. So you can clearly see wherever there is a soil zinc deficiency, the crops grown there are zinc deficient. Means all the grains or the fruits or whatever they are produced, they are zinc deficient. And the people, those who are residing in those provinces, eating these crops, foods, they are also zinc deficient. That means it operates at the system level, from soil to the plants and the food products to the human. So this operates at the system level. And this paper received a very good recognition and also appreciation. So this is one case where, my student, he took it as a challenge, I did it. And he also got a very good appreciation for this paper. And most of the times, the definition, when you look at the literature, there are different definitions of biofortification. So the definition of biofortification is increasing the concentration of bioavailable grain zinc and its promoter compounds are reducing the concentration of anti-nutrients by genetic and agronomic approaches. This is what I'm saying for the grain zinc biofortification, and it holds good for other kinds of biofortification with other mineral elements as well. And wherein you can increase the grain zinc content and also increasing the promoter compounds like organic acids, fatty acids, amino acids, and other compounds. And also you can reduce the phytic acid, tannins, and all these compounds. So all this increasing the beneficial compounds, promoter compounds, and reducing the anti-nutrient compounds, all this process. So finally, making more zinc available for the human body. So the whole process is called biofortification. So you may increase the grain zinc content. If it is not available and if it doesn't make improvement in the health, what is the use? So ultimately, any nutrient improvement should be bioavailable and it should do job in the body. It means it improves the health of the people. And as I mentioned earlier, so for a farmer, yield is very important. So you may increase the grain zinc content, the yield, pest and disease resistance, biotic stressors. It's a kind of portfolio of traits that are very, very important for the success of your biofortified products in the field. And also farmers are the consumers, the end users, they have to like it. The taste should be good, quality should be good. So your biofortified product should be high yielding as well as nutritious for making an impact at the ground level. And as I mentioned earlier, we identified some of the best house lines with high zinc content and acceptable yield potential. So these are some of the Swamy, UCP 122, Waterwala, Kaliboro. These are some of the interesting house succession germplasms with higher levels of grain zinc content and acceptable yield potential. As I mentioned earlier, like for any breeding program, tried genetics identification of donors and using them in the breeding program is very, very essential. That's the basic thing. So at IRRI, my team, we do a lot of tried genetics and also the breeding, varietal development. We identify donor lines through characterization and we develop biparental populations, multiparental populations. We conduct genome association studies and we do a lot of in silico analysis to understand whether these QTL regions or the genes are really useful for a breeding program. I'm not a molecular biologist or a hardcore molecular genetist. I'm a molecular breeder. I'm basically a breeder, molecular breeder. But we are supported by the molecular genetics team. So they help us. But whatever QTLs and genes we identify, we validate through all these approaches and make sure that the QTL and genes what we have identified, whether they make sense in terms of their biology and their functions. With the, as you all know, RISE model serial, a lot of information is already available in silico. So before using those QTLs and genes, we make sense out of those QTLs and genes, which one should be prioritized, which one should be used in the breeding program. And most of these things are done by my students. They have done a very good job and they got very good publications out of this work. And through our genome association studies and also bioparental mapping, we identified two major effect QTLs for grain zinc content, 7.1 and 5.1. 7.1 was identified in magic population, Indica magic, global magic, characterized at IRRI as well as in field rice stations. And this locus is very, very consistent in the Indica and magic populations. On the other hand, as I was mentioning that we also developed two panels of house with different maturity groups, we identified 5.1 locus. So interestingly, when we tested these two locus in most of the elite breeding lines and the elite varieties, the positive alleles for 7.1 and 5.1 were missing in the elite breeding lines and the varieties. This clearly shows that during the green revolution, while improving the varieties for yield, most of these beneficial alleles for nutrition have been left out. And before using any or gene in the breeding program, you need to validate it, whether it's stable across the genetic backgrounds, whether it's consistent and different environments. You need to validate it first. So these 5.1 and 7.1, you can see we validate in multiple populations, biparental, multiparental, and magic populations, very, very consistent across the populations, environments, and genetic backgrounds. And we wanted to see what is the relationship between this 5.1 and 7.1. Just this is the kind of curiosity always that motivates a scientist. Search more and do more and find out what is the cause relationships and how it's really useful for developing products. When we looked at the relation between the 5.1 QTL and the 7.1 QTL, the GIP5 and GIP9 are the candidate genes for 5.1, and the 7.1 candidate genes are nucleotides synthase genes. When we look at these two locus, it's very interesting that the immediate neighbor genes that are influenced by the 5.1 genes, GIP5 and GIP9 are the NAS genes. So it's very interesting. This is the master switch. GIP5 and GIP9 are the master switch, and they influence the expression of NAS1 and NAS2, these NAS2 and NAS3. So it correlates. So if you have both these positive alleles for this transcription factor and these genes, we will get better genetic gains for grain gene content. And also you look at the expression of these genes, you can see the red color in the panicle, in the root, in the endosperm. It gives us evidence and proof that it is working in the metal homeostasis pathway, and it is related to zinc uptake and loading, metal uptake and loading. It gives us proof and evidence that will strengthen our case of our QTL for mobilizing them into the breeding programs. And just to see, we may identify a lot of QTLs. There are certain QTL combinations works better over the other combinations. So you can see with the increase in number of QTLs, the grain gene content effect goes up. The lines with 4 QTLs have a higher level of grain gene content up to 17.7 ppm. But you look at the 2 QTL lines, there are certain combinations of 2 QTLs, they perform better over the other 2 QTL combinations. So when we do breeding, so we have to take into account this kind of interactions between the QTLs and QTL effects. And this is done by another student, Descal Sota and Kalayugan. They did a very good job, very innovative students. And nowadays, a lot of discussions around haplotype-based breeding. You may identify a QTL or a gene, but there may be better alleles available in the gene bank. As you all know that rice has been sequenced. We have 3000 rice genome sequences. If you identify a QTL or a gene, you can search for the better allele in those 3K genome panel, and you can use it in your breeding program. Rather than just sticking to your multiparental population or biparental populations, once you get the information of those QTLs and genes, for example, OSNAS 1, 2, 3 and GIP genes, you can go back to the gene bank and search for the better alleles. Those alleles give you better effect over the may give better allele effect over the alleles that's available in your population. So you can see here for zinc 6.2 and 7, there are different haplotypes AT and GC, and you can see yourself there are certain haplotypes, they are better values over others. So such haplotypes should be searched from the gene bank and should be used in the haplotype based breeding program. Not only haplotypes, even haplotype combinations can be searched, and suitable combinations should be exploited in the breeding program through genomic assisted breeding program or your backrest breeding program. So you can use it in your breeding program. And for any researcher, you need not start from scratch, especially in a crop like rice. There will be already plenty of information available. First, we need to know what has been done so far. Then once we get a good understanding of what has been done so far, then that's where you need to start building your plans and ideas, research themes. So for grain gene content, so we collected all the available literature related to QTL identification. There are 26 studies with using high density maps. So they actually mapped more than 155 QTLs. From them, we shortlisted 147 based on some criteria. And when we did meta-analysis, we found 52 meta-QTLs. Because these 26 studies are done in different genetic backgrounds, different environments, different countries. So these are from different places, different researchers have reported it. So if some genomic regions are repeatedly identified from across the studies, that means they are consistent. So I need not to do and check everything and start validating. If such information is available already online and diverse teams have identified across the globe, we can make sense out of it. And if you identify such stable genomic regions and shortlist few of them and further validate and use it in your breeding program, it will speed up your breeding program. So from almost 147 GQTLs, we identified 52 metaQTLs. MetaQTLs are nothing but these are the consistent genomic regions where people have identified, more than 2 studies have identified the same QTL regions in different genetic backgrounds and different environments. You can see there is horizontal bar that shows there are some genomic regions. You can see QTLs have been flooded there. There are so many QTLs. So those are the consistent genomic regions. And finally, from these 52 meta-QTL regions, we shortlisted 18 metal homeostasis genes. Means these genes involved in mobilizing nutrients from the soil, root uptake, and translocation and loading into the grains. And for these 18 metal homeostasis genes, we did again in silico haplotype analysis. And also we, as I mentioned earlier, we had 2 panels of house that we field evaluated. So using that field evaluated gene phenotypic data and this haplotype data, so we did a study to see which haplotype does better. It's not just in silico, and we validated the results in the field level. You can see there are genes and there are certain haplotypes, their effects are better over the others, and also haplotype combinations. And also you look at this expression diagram, the red one shows highest expression. These genes are expressed in different tissues at different plant growth stages, and it correlates with metal homeostasis pathway, how nutrients are taken up, mobilized, and loaded into the grain. These are all the studies that give evidence and proof that the genetic studies that we do are really working and useful for the breeding program. And earlier, the breeding programs were happening in a parallel. For example, nutrition breeding program or biofortification breeding program was running parallel to the main breeding programs or the pipelines, like irrigated, rain-fed, and all these breeding programs were running in parallel with the biofortification. But few years back, the thought process started that nutrition should be made must, whether you breed for whatever pipeline, whatever geographical area, whatever ecosystem you breed, or the pipeline that you breed, grain zinc content should be a must component. So then started the process of zinc mainstreaming. Zinc mainstreaming means making zinc nutrient a must trait in all the future rice varieties. For that, we follow the scheme like allied germplasm characterization, allied germplasm enhancement, selection, and mainstreaming. These are the steps we follow in the mainstreaming process. That means to make sure it's not only just yield, all the future rice varieties should have enhanced levels of grain zinc content. And these are the countries where we work on a zinc biofortification breeding project. We have already released varieties and also testing high zinc rice varieties. So in different countries. And as of now, currently we are running the high zinc rice breeding project at IRRI. And varietal release is very important process to get your product with the trait intact. Different countries have a different criteria to release high zinc rice varieties. So some people use a differential, some people use absolute values. So I am proposing this method. So like a global standard for releasing high zinc rice varieties to really make impact at the ground level. And in addition to that, my team, we also work on combining high zinc rice, high grain zinc, high antioxidants, and low GI or diabetic, rice or the low GI trait into rice varieties. And we are developing healthy rice magic and NAM populations. And one of the students working on this project. And now until now, whatever I said, mostly they are all conventional breeding work related to zinc biofortification. Now let's move on to the golden rice part and the transgenic part or the genome editing part. Vitamin A deficiency is a major nutritional problem and it leads to blindness, eyesight problems, poor cognitive development. Almost 190 million children, 19 million pregnant women are vitamin A deficient. So it's a pressing problem. But there is no variability for vitamin A in rice. So when there is no variability in the rice germplasm collection, so you need to bring that variability some other way. So that is done through transgenic approaches. They use the gene from maize, phytoen synthase, and phytoen desaturase from the soil bacterium, and they developed a construct, and they did genetic engineering to develop golden rice donor lines. That was done in the Cabernet variety. Cabernet is a US variety grown in the United States that is not adaptable to Asian conditions. What he did here at IRRI was that we introduced the golden rice gene from the Cabernet into popular rice varieties of Asia. In addition to that, just to get any variety, transgenic variety released, there will be a lot of process, and we have to maintain biosafety regulations at every step, whether it is at the lab level or the screenhouse level or the field level. It is monitored by the regulatory agencies. So everything is strictly supervised. Every seed that you produced, every data that you generate, every facility that you use, everybody that works in those facilities, we have to maintain standard records. It's a lot of work. So for this Cabernet variety, the golden rice variety from US, we used it as a donor. And we introduced them into popular rice varieties of Asia. For that, we need to get food feed safety approval first. For the food feed safety approval, you need to generate lot of data, hundreds of studies, where the gene is inserted, what kind of proteins it codes, whether it's safe, what's the enzymatic activity, toxicity studies, so much studies, so many studies, how it performs on the field level, what is the nutrient component, and the stability of this. So there are so many hundreds of studies you need to compile and submit it to the regulatory agencies. A wider panel with a lot of diverse experience, they evaluate the food and feed safety. Once they get food feed safety, so people can consume it, but still they cannot grow it. So then after that, we have to process the commercial application. Again, commercial application, we need to produce a lot of data, and the appropriate bodies, they take a decision. In the Philippines, we got both food feed safety approval and also commercial cultivation approval. And at the same time, we received a food feed safety approval. That means golden rice can be consumed, eaten in USA, Australia, New Zealand, and Canada. And also we have submitted application to few more countries. They are under process. And as I mentioned earlier, we used a Cabernet GR2E variety, and we introduced them into popular rice variety of Philippines. PSB RC82 is a popular rice variety from Philippines. BR29 from Bangladesh. IR64 is popular in Asia. We introduced them into these varieties, and RC82 introduction lines has been successfully released for commercial cultivation in the Philippines. We have also introduced to more than two dozen varieties, so of Philippines and other countries. So some of those lines are being tested and will be submitted for variety release process. We can see the process, for every step in transgenic activity, how we strictly monitor and conduct our studies. And for golden rice or any transgenic activities, always there will be a lot of interest. They ask a lot of information, whether it is same as any ordinary rice varieties, does it have any increment in any other compounds? So like that, so many things. So when we did the nutritional analysis for most of the compounds that are there with the ordinary rice and the golden rice, they're almost similar for almost all the compounds, and they are within the biological limits, safe limits, except with the addition of beta carotene. So it's safer for consumption and healthier. And always people used to ask us how it tastes, smells compared to the regular rice. And once we got the food feed safety approval, so we did quickly the taste and smell test. And this is William Darr, the former Director General of ICRISAT, and his team. You see, they are tasting golden rice, and you can read yourself their opinion after eating the golden rice. So they said that it tastes, smells just like any ordinary rice, and even they said golden rice is much tastier. And golden rice been commercialized, and now we are deploying it in the Philippines, in different provinces of the Philippines, wherever we find vitamin A deficiencies in a bit more problem. So we already selected those provinces, and last season they grow golden rice in 40 hectares, and we harvested almost 40 tons, 100 tons in 40 hectares. And that is being used for promotional purposes, and also the second crop is in the field. And you see, we are distributing seeds to the farmers, golden rice seeds, and you can see I am there in the field, farmers field. Fantastic crop, very good yields, and very clean and neat fields, and very good crop stand. And you see, I am tasting golden rice myself with our team. So now finally, golden rice is with farmers and the consumers. Yeah, and almost 100 tons of golden rice is produced, and it's being distributed for the promotional purpose and consumption purpose, and also used as a source seed for the next crop. And overall, golden rice gene is genetically highly stable. Molecular analysis shows it's clean, neat event with agronomic characterization shows that they are just like any other ordinary rice varieties in terms of growth, development, agronomic characterization, nutritional value, and environmental safety. So overall, it's a golden rice is safe. And for the high iron zinc, so at IRRI, our molecular genetics team, they are developing high iron transgenic events and also genome-edited products. They're using genes related to iron storage, iron translocation, and loading, and also a combination of these two approaches. And they have developed very good events, and they have been tested at the field level in the controlled conditions. And we have shortlisted 2 events, lead and backup event, and we are introducing them into popular rice varieties. And these are some of the events of NAS ferritin gene transgenics. You can see the levels of iron and zinc, almost up to 15 ppm of iron and almost 45 to 48 ppm of zinc in these events. These events were tested in IRRI as well as in C at Columbia. So they are performing almost same in both the locations and with higher levels of iron and zinc. Now golden rice has been released. So the next target is to combine all these nutrients like iron, zinc, and vitamin A. We are stacking them into individual varieties. So already we started this project, and it's for the next phase. And we in these varieties you will get iron, zinc, and also vitamin A simultaneously in the same variety. And this is during the 1960s when we were a food shortage, and we were going around the world asking for food. So this is the slogan by Norman Borlaug, Nobel laureate, who got Nobel for his contribution to the agriculture sciences. So food is the moral right of all who are born into this world. That was the slogan in during those days. So now all the scientists globally, they work very hard, and they made sure that we have enough to feed our people. So now we can say that it's not just providing the food, it's also a duty of our generation to provide nutrition as well along with food. So now we can say that nutritious food is the moral right of all who are born into this world. And this is our team. So really they are all working very hard and very innovative people. I would like to thank all of them for their contributions. And this is the team, IRRI-wide, and also our partners across different continents, different parts of the world, students, and everybody, all my team. And also I would like to thank generous funding from Gates Foundation, USA, IRRI, Aerospace, and Taiwan Koei. So many people have organizations have generously funded our work. So with that, that's my last slide. Thank you very much for your patience and listening. Thank you.
Thank you, sir. It's a great talk. Many appreciations is there, many questions also there. So we already collected all the questions and give participants for more time to ask you questions based on your talk and also for your from your interview. So we prefer to go for the interview round now, and at the end we will discuss and questions answer round.
Fine with me. Yeah.
We collect many questions. I will send all the questions to you by the mail, but for that timing, we select 4 or 5 questions, which is very relevant to your work. And uh, as, as you know, our audience is from graduations to scientists, so variety of questions. So we prefer the questions from the young minds. Those are have some uh, basic questions on biofortification and all. Shoma, please
That,
go for the interview
that,
session. Yes, sir.
that's fine. That's fine with me. Yeah.
Okay, okay, sir. So go for the interview. Sir, this session is a motivational session for us. As you know, the young minds are there. So we love to learn your journey. Please give us some guidance, some information, some takeaway message during this session. So people are work on that, and I think I'm sure they will benefit it in the future as you
It's my
develop.
it's my
Soma,
pleasure. Yeah.
yes, sir. Thank you. Soma, please. And participants, please put your questions only. I know you have many suggestions, like topic suggestions. People are asking for different plant, different approaches, nanobiotechnology, and all of this. We have a special column in feedback form. Put there. I'll go all the your suggestions. Try to find out the speakers, and definitely in future we'll go for that type of topic, in try to find out that type of scientist. Those are specialists on that topic. So put your
So,
suggestions in feedback. Shoma,
shall we stop the screen sharing for now?
no, yes, sir, please stop your screen sharing so we can, yes, okay. Shoma, go for it.
Okay. So you were watching Dr. Swamy, and he was giving a wonderful talk on biofortification. So now we will move forward towards our interview section, which is very popular in Bioengine, because all the viewers, they know, they see, they read about the research, but they would love to know an up close and personal to with the scientists and what they think about a few things. So, sir, we'll start with the session. My first question to you will be that we would love to know about you more. So, we would love to know how you built your career. Could you please share your journey as a plant scientist with our viewers?
Yeah. Thank you, and thanks for this opportunity. So just like you all, we are also just ordinary people. Started our career at the bottom level. I'm also from a farming family. I also grown up in rural areas. So I also got education like all you people. But at certain stages, you need to take challenges. So you need to make decisions. And in this process, you are not alone. You will be guided by many people. So I am lucky that always I got very good teachers, to be frank. So always I got very good teachers. They always directed me. They given the path. So that's how whenever I had to make decisions, I have to consult people. So sometimes we think what we know, that's the best. Always it's better to interact with people, learn from the experts, and get their advice and make a career. There is no field, every field has its value. It's not like that that science is better, this science is better. It's all about your interest, your passion, your passion. You can make a with your creativity, with your ideas, with your thought process, you can build career. You can make new contributions in every field. It's all about your interest and your passion. So and also it's not like you get all. Sometimes we hear that if I get that facility, if I go to that lab, if I go here and there, I can be a better scientist. You need to start somewhere, and you need to show that the passion for that. So people always recognize you, and also you should be ready to take challenges, risks, and always everything will not come up with best results. Always there will be failures. Always there will be success. So a combination of things. But I always took it as a challenge and determination. So in whatever we do, we always think that. So don't give up. If some people say something or they say, oh, it doesn't work, you know the best. Just to give you one example, my teacher always, he was a very strict man, but whenever I was going for giving a seminar or a thesis defense, he used to tell me, you know the best, you know better than the committee members, be confident and you present it. That's the kind of motivation he used to give. And also at the end, as I was saying, contributing to the food security is a lot of satisfaction. Okay. We touch the people, real application, providing food and nutrition and better crop varieties that can make changes at the rural level, at the ground level.
Yes.
So that is more satisfying. So these are all the combination of many things in life. It takes a lot of twists and turns. We learn, we fail, we succeed. So we absorb everything and we need to move on. That's all what I say from my side. Yeah.
And on that note, what motivates you most to work for the betterment of agriculture?
As I said, when you touch the real consumer target people, when you develop better crop varieties, as I said, food and nutrition security is most challenging. And just like me, there are several thousands of scientists across the globe. They are working very hard. So we are providing food and nutrition through crop variety. They have done a good job, all our predecessors. Just our contribution is very little. And that motivates us. The most exciting part is that when you conduct an experiment, when you see the result, when you go and check the result in the field, in the screen house, when you find something new, it's always exciting. And also when our students, when our stakeholders, if they feel happy with our products and our results, our experiments, our outputs, it's more satisfying. It motivates. So these are the even one who is really involved in these kind of things. If they start looking at everything minutely and start enjoying the process, always you will keep motivated. You don't find time to think that I did not get that. It's not happening like this. Once you are involved, every second, every minute. For example, I cannot. In every day, I have to see rice plant. I go to field at least once or twice to visit the field. So always is a kind of passion you need to develop. Yeah, that motivates you when you see all your results and your students growing up and they becoming bigger. This thing when you present it, when your products are in the field. So all these kinds of things motivates us. Yeah.
Right. So. Do you think biofortified crops will play a major role to fight against malnutrition?
Yeah, as I mentioned in my presentation, that is the reason, that is the most cheapest and sustainable way of providing nutrition. So to wider range of populations and those who are vulnerable populations. So this globally there is awareness and interest to develop nutritious crop varieties. That's why they are investing a lot. That's also the reason the zinc mainstreaming project is happening, and also the characterization of heirloom rice. A lot of diversified products are available in the market. If you go to supermarket, you will find all kinds of rice. I zinc, I nutritious. So a lot of red rice. That so many things are happening. Lot of awareness is there. That is the reason biofortified rice varieties are being developed, and also they are being made mainstreamed to make sure that nutrients are available in the food they eat. Yeah. Okay. Since it's cheaper and it's like any other ordinary rice, they need not to invest extra money. So it is the sustainable and cheaper approach. Yeah.
So golden rice is now available in the Philippines. So do you think that other Asian and African countries will follow this example of Philippines and do the same?
Exactly. So always somebody has to take the lead. So Philippines has shown the way as their leadership and the system really working well. And we are targeting other countries in Asia and Southeast Asia. And also in the next phase of the proposal, we will be moving into Africa as well. If everything goes well, there are selected countries in Africa where we will be taking up this project. Yeah.
Okay. So, what is your opinion on GM crops, and do you think they have a future in attaining food security?
Yeah, already they are playing important role. You might those who are in a transgenic area, and there are some certain websites every week they publish how many crops are being transgenic crops are being released in different countries. You take the case of US, Australia, New Zealand, Canada, wherever the science is more progressed and they have acceptance. So a lot of crops are being released every year. Even in Africa, cowpea, even in Indonesia, even you take brinjal in BT brinjal in Bangladesh and Philippines, there is a lot of acceptance. And moreover, now genome editing is coming up in a huge way. So definitely there will be countries. So those who have acceptance for these kind of products, they are going in a huge way, and these kind of crops are being released. Yeah. And once there is a once the path is shown and they are good and good for consumption, safe for environment, human health, and everything, these are actually rigorously tested before being released. But there is always some amount of perception issues. But as a scientist, we have to show that everything is clean, safe, and everything proven. Then why not? Yeah.
Okay, so what is your opinion on digital agriculture, and would it be wise to combine artificial intelligence and agriculture?
Yes, already it's happening. Digitalization happening at every level. So from research level and product deployment, product development, and product production, consumer side, marketing side, at every level, digitalization already happening. And that is going to happen. Even all the mechanization is linked, all your drones, all your sensors. Yesterday there was a news you might have heard. Now they are developing artificial honeybees kind of stuff. So they want to increase the pollination in the field. So the miniature honeybees, artificial bees, they want to release it in the field to increase the pollination rate, so they get a better yield. Like that, a lot of things are happening in terms of crop management, like your water application, fertilizer application, and all those things, and also your data handling side and data analytics side. And also farmers, just farmers nowadays, they don't go, they use just mobile to switch on their electric pump, motor pump. Like that, a lot of things are, and also farmers get the fertilizer management options just in their cell phone. Just they give what is the kind of soil and all these things, they get it. And also digital soil maps, and also for crop insurances, and for climate change issues, and also classifying the environment into different ecosystems. Lot of things already happening, and it is going to happen in a huge scale. But as I said, from in the whole value chain, whether it's rice or any other crop, it's happening in a huge way. For example, us in IRRI, for as a scientist, all our data collection, we don't use any more paper and pen, all gadgets. And in the field itself, just you click with internet, it is already uploaded to your database, like that. And big data analytics. So in almost all the field, crop development, crop production, and whole value chain, all the consumers, and also you see the blockchain technology. From the produce until it reaches the consumers, you can trace your product. So whether the right product that you produced has really reached the consumers, like that. So much of digitalization is happening, and artificial intelligence is being used for all this process based on all the informations available, algorithms available. So everything is being integrated with machine learning and artificial intelligence. It's not just one aspect. It's very diverse field, and every small area and small aspects, they are trying to make use of all the artificial intelligence information. Yeah, and also in addition to that, maybe in the next question I will answer that. The younger generation mindset is also changing. So the kind of work culture and everything in the next few decades may also change. So the whole world has to be ready for that.
That was amazing. According to you, which sectors of plant science research do you think will prosper in the near future?
For me, every field has its own value. It's not just people fancily they choose a few sectors and they say that. Ultimately, to make an impact, it's the coordinated work of all the parts of the plant sciences. Okay, different aspects of the diverse subjects of the plant science. They have to work together. For example, in rice, just to give you a breeder, a molecular biologist, and a soil scientist, water management scientist, economist, we work together to make impact. Because these words like genome editing and all these things, so hi-fi words, just because of that, it's not the frontier science. Ultimately, it has to make impact at the ground level. Everybody has to work together. So, but with respect to this one, I can say that this genomics assisted breeding and the big data analytics and also genome editing, these are the few areas in terms of rice development side. So some important things, but every field has its own value. Even its extension specialist, those who communicate the information on the marketing side, lot of things are there. So every these aspects of the science, interesting fields, just to know what your consumer wants, what they eat. There's a tremendous interest in that area, and how to market your product innovatively. So there's a tremendous interest. So a lot of fields, every aspects of the plant science has its own value. So it's your interest. It's your interest. So you choose it, make impact in that area. Yeah.
Right. So now let's talk a little bit about the scholars, the younger researchers. How should a scholar handle long periods of experimental failures or unsuccessful events?
So first thing is that don't give up. So even failure is also a result. So we need to learn to accept that failure is also a result. Now there is a lot of discussion around that. If somebody failed, you can publish that as a thesis, as a paper. So that other people not to follow the same thing. So you try one, you did not succeed, you try something else. Don't give up hopes, and also don't get discouraged easily, and also don't switch topics so quickly. So as you invest time, so always I tell my students, when you come up with a problem, how do you solve it? Just I ask them, so how do you solve it? So they say, so first you go to the literature, search, find, read yourself. Then you don't find solution, discuss with your friends. Then if you don't find, you go to the experts. You need to follow a sequence, not to jump so quickly. So you need to give time for your brain to think and process it. You will always find a way. So failures means don't give up. So be positive, and always okay, choose once you are 100% sure and you are convinced, then choose a different path, different fields, but don't give up easily, and make first all homework, contribute, work hard. And suppose if you are successful, after you think that you have chosen a area that's so difficult, so challenging, and the others have already given up, but you have chosen a field and you succeed at the end, you will get most appreciation. People will appreciate you. Challenging, big challenges, more appreciation, you prove it. So don't give up so easily. Trust yourself, be confident. Why it cannot be done? Just always think why it cannot be done. Just think, and be positive, motivated. That's all. Even failure also, as I said, you can publish that. It's not like that everything that you should end up with a rosy picture and very good products. So that kind of mindset also should change. Yeah.
Right. Sir, like when students or when scholars or young researchers, they start professional collaborations, they are sometimes apprehensive, or what would you advise? What qualities and concerns should they look out for when they start a professional collaboration?
Yeah, for the collaboration side, you need to see what you can offer. So, and also you can see what mutual benefits and how you can complement each other. Okay, and how your teams works mingle well, and how you can make an impact at the end of the day. And at the same time, you can have a background check, and you can see what groups are doing what is their contribution. Always there will be publications. You interact with them, you learn about them. It's always a mutual kind of thing. So what you give and what you take, and how you can complement each other, and how you can make if you work together, how impactful it will be your work. If you work, you can make certain level impact. If you both of them work together, your impact will be much better. So, and what kind of deliverables you can make, how you can share and care, how you can save your resources, how you can use your skill sets. That group may have certain skills, your group may have certain skills. If you combine it together, you can make much more better science and better delivery. And always be respectful to others' ideas and thoughts. Okay. So any idea, no idea is less. So every idea is its value. Be polite, accept it, and listen more with patience. And so collaborate with respect. So always there will be a fruitful collaboration on that.
Sir, I'm sure you get a lot of requests from aspiring researchers to, that they want to join your lab or so, or start a training or whatever. So when you hire a researcher or a fellow, what qualities do you look for other than academic qualities?
The first thing I ask them is that, why did you apply to my group? So I ask them, can you make a statement of purpose? And I see their interest. So sometimes for the second mail, we don't get any reply after that. There are some, they respond. So that shows the commitment, and also everybody wants to grow. That is always there, but they have to make certain preparations. So before you approach or you want to apply somewhere and ask for opportunities, first you have to assess ourselves, what we are. So you have to go to certain groups and work with certain people. So what is my ability? What is my what I can contribute to the team? It's not just like that group is doing good, I can go and join them and do some work. You need to think what you can contribute. So that's the thing. I see their commitment, and also the way the scientific capability, the way talk, the way interact. So in addition to science, the equal thing is that we do teamwork. So you have to mingle with the team. So in my team, we have so many people. They have to work with all of them to get your work done. Collaborative work and your commitment and also passion for science. So when we interact with them and when we talk to them, already we can guess. So with the kind of answers they give and all the SOPs and also the proposal, and also I ask them once they make a SOP, and I ask them make a mini proposal. So, but always I give opportunity to as much as possible some unknown people, new faces from new labs, new groups, and also some students really brainy, but they don't have kind of they will be having a very good theoretical background, but they don't have facility to execute at that level. Maybe in the small labs and small environments. So I see them, if you give them opportunity to them here, so definitely they will excel. We see so many things, but sometimes, most of the times we succeed, and also we also fail sometimes to choose. Yeah, that is also there. Right people.
So we are at the end of this session, and we will end it on a positive note, motivating note. So what would your words of advice be to the students and scholars who are new to the field of plant science research?
Yes, so you are in the right field. So because you have chosen it, because you have some interest in this area, you have lot of interest. You have passion for this. So do whatever level, whatever facilities, everything, whatever is available, do your best. Keep motivated. Never give up. Be determined, and always be creative. All work as a team. Okay, there are certain times your colleague may be doing better. Sometimes you do better, but be in a team. Enjoy the work and work as a team and deliver and do your best. As I said, every field of plant science is good, and you can make an impact. If you are creative, you can find a way. There is nothing like the thing, everything. Sometimes students ask me, sir, there is no more thing to do in that field. Already a lot of things have been. No, that is your limitation, not the science limitation. You need to think. You need to think more. So always there will be new ways, new pathways. Those things emerges. Be determined. Don't be discouraged by comments and suggestions. Be positive. And do your best in your field. and be good to others, be a good listener. Yeah.
Okay, so we will go back to Shovu and see if he has questions for related to today's talk. Shovu, shall we start?
Just a minute. For Vijay, are you there? Yes, Vijay is there.
Yes,
Sir, actually, I wanted for 3 minutes to introduce Vijay. Vijay now joined Bioingene in this January 2023. Actually, he is also a breeder and he did his PhD from PK Gupta, sir. So I'm very excited, and we are recently meet in a seminar, a conference, and we talk about it and feel that he has also interested in same intention also to educate people to spread plant science. So I invite them to join Bioingene, and it is his first, and I wanted to introduce because now we are going to the end of the session and certificate distributions. So to hold up participants, this is the right time to introduce him. Soma, please introduce.
Yeah, nice to see you, Dr. Vijay, joining this team.
Thank you, sir. Excellent lecture presentation, sir.
Your team is really doing a great job. So
yes, sir. We are working on wheat.
yeah. So all the best for your team. Yeah.
Thank you, sir. Thank you, sir.
Soma, please introduce Vijay.
So we have a new member in our team joining Bioingene, and we are very happy to welcome him. We are welcoming Dr. Vijay Gahlot. He is working as an assistant professor at University Center for Research and Development, Chandigarh University, Mohali, Punjab. Earlier, he worked as DST Inspire faculty in CSIR, Institute of Himalayan Bioresource Technology, India. With 12 years of research and teaching experience in areas of crop biotechnology and genomics, he has identified several genomic regions and candidate genes on wheat chromosomes that were involved in water and heat stress tolerance. He published over 45 international research papers in reputed journals and several book chapters. He is serving as editor in nearly 10 scientific journals. His Google Scholar H index is 20 and I-10 index is 24. Recently, he was inducted as a member in National Academy of Sciences that we know as NASI, India. We give him a warm welcome to Team Bioingene, and we are sure his presence will make Bioingene stronger. Welcome, Vijay.
Thank you so much for nice introduction.
Thanks, Vijay, for coming. Let's do some work together. As you say, is that we are in the right track and right path. And thanks, sir. Now we, Shoma, go for the YouTube questions. I am already highlighted few questions. And sir, few basic questions are there. Do you have any position for postdoc? Few people are asking that questions. And one interesting question is there that how they can get some seeds of golden rice. 3-4 people, 3-4 participants already asked these questions, how I can get some seeds of golden rice. So interesting that people are diversified, different countries, and few people are asking on different questions, not on the rice, others will. I will collect all, Vijay and me will collect all.
So shall I just go on as it is there?
Go for the direct highlighted questions only.
Okay, so Vipina V asks, in India, there are many security policies for GM crops. Is it also applicable for biofortified crops?
Yeah, it depends upon the kind of biofortified product. For example, if it's a GM biofortified product, definitely it has to follow all the GAC regulations. But if it's conventionally bred high zinc rice, then or any other nutrient, it need not to. Just it will follow the ICR guidelines and will be released. Yeah.
Okay. Next, Niaz Hussain asks, very informative, but instead of traditional breeding for healthier rice, why not promote the activity of zinc and iron controlling gene activities to improve the nutritional level of rice?
Yes, that's what they are doing in our transgenic work. So they are choosing right kind of promoters and the genes, and they are increasing their activity so that they are getting expressed in the endosperm, and that's what they are doing in even genome editing. Yeah.
Okay. Abhinav Chakraborty asks, does the high zinc variety have any effect on the production of rice? How is it compared to normal high yielding variety?
Yeah, so when you develop any biofortified variety, so there is a guideline. It should perform similar to any other high yielding variety that is available in the market. Okay, that's the main. Otherwise, they don't. There is release guidelines. So in every country, they have a criteria to release a variety. We need to satisfy that before getting it released. Yeah.
So Dhan Stephen Mandin asks, have you also considered using specialty rice like pigmented rice for biofortification, since they are said to have higher zinc and iron content?
Yes, definitely. That's what I mentioned, the heirloom rice project. So, but at the same time, you need to consider, just they have a nutritional value. At the same time, they also have a high anti-nutrient properties. For example, phytate content. So, you need to choose right germplasm, and you can promote it in the special market segment, not as a variety, because their yield levels are not that high. Otherwise, you improve their yield level and maintain the nutritional value. Otherwise, you can promote them in a special market through packing and selling it in the supermarket. That's a special market segment. But biofortification, it needs to be in the popularly grown rice varieties and high yielding and should be high in nutrition. Yeah. That is also for heirloom rice. There is a lot of interest nowadays, as I said. Yeah.
So there are 3 final questions that I will ask in tandem. You can choose to answer. Can I get some golden rice seeds? Next, sir, is there any postdoc position in your lab? And lastly, any information about golden rice developments in India?
So for the postdoc position, you just keep looking at the website, IRRI website. Okay. So you will find whenever opportunity comes, you will find that. Okay. For the golden rice, other than Philippines and also Australia, Canada, and New Zealand, so you cannot eat it. So and you cannot transport it from Philippines outside. So if you come to Philippines, you can eat it. For the Indian government, as you know, there is still a policy, regulatory things needs to be improved or updated related to transgenic works. And we had a project with the Indian institutes developing golden rice. There was, but in the second phase, there is not much collaboration on that.
Thank you, sir. It was a wonderful and very informative session. And let's go back. Yeah.
Yes, sir. And as you guess that how many young minds are there right now. So many people are asking you suggest PhD topic. so that is the reason, and we appreciate these questions because now we are connected to right minds, because now we are in that situations we can change the topic also. So it's a right time, and please anybody can say the topic in that way. It's totally depend upon the situation, institute, and your PI experience and availability. Topics should judge, should select based on this criteria and your intention. It's not like that AI in agriculture is booming, so AI CRISPR is booming, so CRISPR not there. Choose a problem. I will suggest choose a problem, not the technique. And technique may change, but problem is very important for that.
Completely agree with you.
Yes. Thanks, sir. And sir, it's a really good webinar, so interactive. I actually, sir, I can't afford the Zoom webinar. Otherwise, you can see the actual people in right here. Many people are there. All are commenting, all are watching. I'm collecting all the questions from there, from YouTube live.
But Zoom webinar is also limited.
No, you can buy 10,000 participants, but it's too costly, and so it's a one way to connect and build the maybe in the future we can try this. And sir, this is a great webinar extension and achievement. You have achieved a lot, and we wish you go more and more. And like Bangladesh is planning to for the golden rice commercializations. We hope they did it, and then many others country also going for that. Biofortification is important. Right now, few people are asking about India. Yes, in India, normal breeding means conventional breeding, biofortified, 14 varieties recently released in different zone of the India, part of the India. And rice biofortifications actually is a successful story of what I can say is a metabolic engineering. So it's inspired us, and we are very happy that when we are students, we also, because it took 20 years due to these regulations and all, when we are students, we basically myself, I'm few things attract me for the field. One is a golden rice. So I'm very happy that today we can bring someone who are actually working on these aspects. Thank you, sir, for coming, and thanks Vijay to join this team. Thanks Shoma for participation.
Should you not say about the next webinar where we are planning to bring?
Yes, we are actually. I'm putting the information when it's finalized. We are planning a special webinar for International Women's Day celebration.
Science, yeah.
So where we are planning to bring 4 or 5 women scientists, 20 minutes talk and 30 minutes discussions, where we try to discussion not actually plant science, where we mainly there will be a talk for the plant science, like opportunities in plant science for the graduate and undergraduates for the different fellowship and all. There will be a talk for the how to set up a lab, how to apply for funding, and like that. It's not finalized yet, but maybe it's 9th of March at around 3 p.m. We'll promote and we'll share all our viewers. And next will be a webinar on agri-tech. Many people are asking for the agri-tech system. So in ICR Institute, a director agreed to that, but I'm not getting the date, but it will be in the March. Sir, thank you. Thanks for coming.
Thanks as well. And also, I had an opportunity to interact with so many people, and also I also want to wish you success in your field and also in this bioengine. So thank you very much.
Thank you,
sir.
Thank you.
Thank you.
Vijay, you please stay for 4 or 5 minutes. Sir, thank you.
Okay, thank you.
So much stop the broadcasting. Thanks every participant. Just I'm going to give the link for today's certificate applications.
Okay. I'm stopping live streaming now.
I don't know. I need to check it.
I am stopping it. I have not yet stopped since you said something. So
okay. No. You can stop. You can stop.